Hello, I'm Marc Sharp. I am the Chief Scientific Officer at Linnaeus Bioscience. We're a company that uses some proprietary technology to study the mechanism of action of new compounds in a wide range of bacterial species. And Recce approached us a little over 2 years ago to start looking at the mechanism of action for Recce 327. And it's been a very exciting compound. It's got a unique mode of action that we've not seen in any other compound before. So I'll take you through some of the results we've seen and what that's telling us about what Recce 327 is doing. Recce 327 has a very broad spectrum of activity. So it's active against both gram-positive and gram-negative bacteria. It also affects different mycobacterial species. We've tested it against a subset of the escape pathogens, Bacillus subtilis, which is a gram-positive bacterium, and Escherichia coli, both in biofilms and also in just free culture. And it's effective against all of these different species in different conditions. I'll be going through a series of graphs coming up, looking at both ATP levels and the viable cell counts, which are both ways that we look at how a compound is affecting cells. When we first did testing with Recce 327 using our cytological profiling, which actually is a way of looking at cells on the individual level to see what's going on, the first thing we saw with 327 was that it was arresting basically all cell activity. So the cells were no longer dividing, they were no longer growing, but they were not lysing, which is something that you typically see with compounds that were affecting the cell wall or the cell envelope in some way. So this was a unique mode of action we had not seen before. That led us to start looking at things like cellular bioenergetics and whether that was being affected. And we found that Recce 327, one, affected cellular bioenergetics very rapidly and very strongly, and at the same time was also killing cells very rapidly. One note, we did all of the testing I'll be presenting is in 10% LB rather than pure LB, which is a very rich growth medium that we do a lot of our work in. We looked in 10% LB because there we were able to look at a range of species at the same relative concentration. So the concentration we'll be talking about is called the MIC, or minimum inhibitory concentration. And that's basically the concentration where you see essentially no growth when the cells are treated. And that's just a way so you can compare similar conditions across different bacterial species rather than looking at the pure concentration, which can be a little bit different between different species. So first up here is E. coli, which is probably most of you have heard of E. coli. It's a very common bacterium. It's in the human gut, and it also causes different kinds of diseases such as food poisoning very commonly. On the left here, you'll see a chart of the ATP levels in these cells under different conditions. So treated with no Recce 327, 1 times the minimum inhibitory concentration, 2 times the MIC, and then 5 times the MIC. And particularly with 2 and 5 times the MIC, you see a very rapid and substantial decrease in ATP levels. And then on the right, you'll see viable cells. So this is literally counting how many cells are still alive in the culture. And that graph is actually charted on a logarithmic scale. So it goes from about close to 10 billion cells per milliliter of culture to less than 200 cells per milliliter of culture. So Recce 327 is killing virtually all the cells in that culture within an hour when you treat with 5 times the minimum inhibitory concentration. So then we also did the same, basically identical testing in a different strain. This is Bacillus subtilis. This is a common soil bacterium, and it's a good model for a lot of gram-positive bacteria. And again, you see the rapid decrease in ATP levels when you treat with 327, and then a concomitant drop in viable cells. Again, we look in Pseudomonas aeruginosa. This is interesting. This is a wild type version of Pseudomonas aeruginosa. This is an extremely resistant pathogen. Very few drugs affect this pathogen. And we're seeing, again, very strong effects from 327. And then Acinetobacter baumannii, another gram-negative bacterium. Klebsiella pneumoniae, again, you're seeing the same thing. Rapid decrease in ATP levels correlated with a rapid decrease in viable cells. And when I say rapid, I mean we're seeing a very significant effect virtually as quickly as we can test this. So within 10 minutes of treatment, you're seeing very strong decreases. Staphylococcus aureus. And this is a specific strain. There were some technical difficulties testing with Staph aureus, which is not at all uncommon with Staph aureus. It's a difficult strain to work with. So the techniques we were using for the other strains were not really working in Staph aureus. So we had to use a different one. But again, in this strain, we see a very rapid decrease in ATP levels, and that is also correlated with a very rapid decrease in viable cell counts. So this was all testing in just regular culture medium. We also tested 327 in what are called stationary phase or non-growing cells. And this is very significant because many antibiotics will not kill non-growing cells. Antibiotics such as the penicillins, they affect the cell wall, but it will only kill bacteria if the bacteria are actually growing. And when you have an infection, you very commonly have a population of the cells that are not growing. So if you're treating with an antibiotic that only kills growing cells, it will have virtually no effect on that on the cells in that infection that are not growing. Recce 327, again, kills non-growing cells, and it kills them very rapidly and irreversibly. So that's very interesting. And then similarly, Recce 327 will kill bacteria that are growing in a biofilm. And again, biofilms form particularly on implantable devices, such as catheters or joint implants, and can be very difficult to treat with antibiotics. We use several different common antibiotics as controls. Tetracycline, which inhibits protein synthesis, ciprofloxacin, which is a DNA replication inhibitor, cephalexin, another cell wall inhibitor, and then colistin, which is membrane active. It basically just permeabilizes cells. None of these were very effective against biofilms, but 327 was extremely effective against biofilms. So again, another encouraging indication for this, this compound. So again, in conclusion, 327 is rapidly bactericidal, in largely, if not completely, irreversibly so. Reduces viable cell counts very rapidly. The effects are dose dependent, and it has very broad spectrum. It's bactericidal, and it's effective against both growing and non-growing cells and cells in a biofilm. So this is a very interesting compound, and we've been really enjoying our chance to study it in our partnership with Recce.